the overall vision for my lab, it's actually to create what we call digital twin models in biomedical engineering, and we primarily focus on cancer treatment and next generation therapeutics. But I also wanted to acknowledge the fact that we also have an educational mission, and we want to nurture the best talent in students from very diverse backgrounds, and to catalyze responsible innovators for tomorrow. So the scientific vision in my lab is powered through what I call modern computational science. So this is essentially trying to combine multiscale modeling and data science for biological systems and biomedical engineering. And my lab is well positioned between an institute and a department, that allows us to do just that. And more specifically, the implementation occurs through what we call combining multi-physics simulations at different scales. So here I'm showing you length scale on the x-axis and the time scale on the y-axis, and these are range from molecular level models using molecular dynamic simulations to coarse grain methods to more continuum methods, and more engineering models that involve ordinary and partial differential equations at the larger scale. So we have this vision of combining all of these models seamlessly using machine learning, and then using large high-performance computing in supercomputing architectures to actually run these, models. And where we are going with this is to try and realize this concept of digital twin. And the idea of digital twin, of course, is that you want to represent complex systems, using algorithms, such that these algorithms actually embed the complexity of the biological system, but also include the mechanisms. And at the finest level, one can even include things like patients' individual biomedical and molecular profiles. And so these models will become patient-specific, personalized to each patient. So one vision of this in cancer would be a digital twin simulator in cancer. We call it the OncoSimulator. And this is essentially a heterogeneous multi-scale platform for cancer system models with clinical applications, like I said, with the goal of developing personalized models and adaptation to different clinical scenarios. The other emphasis that we have in the lab is to create a digital twin simulation platform for nanomedicine applications and using nanomaterials, both natural nanomaterials, which I'll talk about today, and also engineered nanomaterials, which I won't have time to talk about. But here we have in mind targeted drug delivery, cancer mechanobiology, and also immune engineering, which was related to the previous talk. Right? So in the interest of time, I'll just, of course, only have time to talk about one of these. And so I chose to talk about cancer mechanobiology. And I'll talk about, in particular, a class of novel materials called exosomes, which are very prevalent. So before I do that, I wanted to lay the landscape of why mechanics and cancer are related, because this is a materials lecture. So mechanics is actually exploited as a screening measure in many cancers, whether it's a mammogram of the breast or ultrasound of the liver. But that's more on a diagnostic level, but can at a more mechanism level, can we actually consider mechanics as a driver? So if you say that there is a stiff tissue, can you actually say that that's what is driving the disease? So it actually turns out that there is evidence for that. So if you look at the mutation rate in various cancers and various tissues, what you actually find is that the somatic mutation rate actually depends on tissue stiffness. And this is actually unbelievably monotonic trend, where with increasing tissue stiffness, you get increase in mutation rate. And this is particularly relevant for tissues like liver, which from a normal liver to, say, a cirrhotic liver, can have up to 2 orders of magnitude increase in stiffness itself, right? So this is what happens at the macroscopic scale. And so the question is, what happens more down to the cellular scale? So one way we can look at that is to do RNA sequencing at a single cell level. So here I'm showing you some RNA sequencing data from liver cancer cells, and these are cells subject to 2 different stiffness conditions. And then once you do this, you can actually then ask, what are the differentially expressed genes? And that's what is shown in blue and red color in here. And if you then go and ask what these genes are, and that's actually shown in the table on the right, you notice that there are several of these genes that correspond to commonly acknowledged pathways at the cellular level. But more importantly, we find cytoskeletal genes being overexpressed or differentially expressed, but also genes related to trafficking, an exosome, and things like that. So we found this very, very interesting. And so the question, of course, was that, is trafficking somehow related to the progression in cancer? Because that's something that cancer biologists never think about. So this is one of the things that actually intrigued our hypothesis. So when I say trafficking, what I really mean are the processes like internalization, sorting in the exosomes, recycling, production of the exosomes, and things like that. So all of these have featured in our RNA sequencing results, which means that they are inherently tied to the mechanics of the soft tissue. And this brings me the concept of exosomes. So one of the trafficking processes that came out are these exosomes and microvesicles. So what are these? So these are particularly topical. So exosomes are 100 nanometer vesicles. They carry cargo containing the host cell. And they are released upon the fusion of the endosome to the plasma membrane that will result in the release of these vesicles. And microvesicles are vesicles that then directly shed off from the plasma membrane. So both of these are present in the extracellular environment, and together we refer to them as extracellular vesicles. But more importantly, they are very potent mediators of how cells communicate with one another. And this is primarily how a cancer cell can be communicating with, say, an immune cell. And has a lot of bearing in terms of the patient's individual immune response. So the extracellular vesicles can pre-program the primary tumor by modulating the immune response, as I mentioned, but they can also impact metastatic colony growth, because these exosomes can travel everywhere in the body, and so they can actually help these circulating tumor cells establish a colony at a distant site. So this is very topical. And so the question then is that why is this then related to the mechanics? Because many times when patients develop tumor in the solid tissues, first there is stiffening of the tissue, and then there is progression to the tumor, right? So that was basically the question. So I'll give you a few experimental results that we have been doing that probe this very unique hypothesis, because nobody's ever tried to put these together. The first is our ability to measure at a single cell level the state of a cell in terms of its mechanical properties. And so one measurement that we did in here is to measure the plasma membrane tension, and we use optical tweezer setups to do that. And every time we've done this measurement, we find that the cancer cells actually have a lower resting membrane tension compared to normal cells. And this was something that was actually quite intriguing to us. But if we then take the same and then put these cells in two different stiffness conditions, we also find that when these cells are subject to stiff environment, they also produce a lot more exosomes, right? So there's something about lowering of this tension and the promotion of the exosome secretion, which is something that we were quite intrigued about. And this is one of the hypotheses that we were trying to pursue. So why is this generally relevant? Because the first thing that we know, and these are some experimental data that shows, so the top example actually shows that when cells are cultured in, say, two-dimensional environment versus more complex three-dimensional environment, the first thing that actually changes is this resting membrane tension. So in the three-dimensional environment, the membrane tension goes down, whereas in the two-dimensional environment, the membrane tension goes up. And that has something, a direct effect on the ability of the cells to survive longer or to die quickly, right? So in two dimensions, when the tension is high, the cells die very quickly, whereas in the three dimensions, when the tension is actually very low, the cells are able to survive a lot longer, which essentially inherently links a very fundamental property of cancer cells, namely cell survival, to a mechanical property, which is tissue stiffness and also resting membrane tension. The other example that I'm showing you in the bottom is the example where if I now subject these cells to mechanically different conditions, there is a lot of productions of the exosomes. And this is very important because there is accumulation of a set of molecules called the glycoproteins, which can rewire metabolism. And so this is telling you that somehow the exosome secretion is linked to a metabolic change, which is very fundamental, again, to the cancer cells, right? So all of these kind of raise some very fundamental questions. And so what we wanted to do is to link the physical effects of the cellular microenvironment, say in a tumor, to the ability for the cells to actually traffic differently and then acquire this cancer phenotype, right? So this is one of the fundamental questions that we want to answer, right? So I'm going to show you some math because that's basically how we do these calculations, right? So we try to model this process at multiple scales. So the first thing that we do is to have a continuum level model. And in this model, we represent the cell in terms of its energy function. And then we are able to then capture various deformations, in the cell. And again, this is powered by some applied mathematics, but also from some computational geometry. So it turns out that it's the same computational geometry we see in cartoons where, you have all these funny shapes. We use similar operators in our simulations. And in the end, what you get is we get to do these types of simulations. So these are Monte Carlo simulations that are developed in the lab that can basically represent the state of this cell according to the different stiffness conditions. So just to kind of give you an idea, there are many proteins in the cell that can fundamentally deform the cell membrane. And if you then ask what are the possible shapes that you can generate from this, then our model actually predicts that these are the possible shapes that you can get. More importantly, if you look in the scientific literature, experimentally, all of these shapes actually arise in these cells. So this is telling us that geometrically what we are predicting from our model is essentially what is happening inside these cell membranes. And so we want to be able to use this to understand specific applications in terms of what's happening in these cancer cells. So we developed a lot of mathematical methods, and one of them is what we call this free energy, which tells you the stability of these structures. So I'm just going to give you some results and some examples. So the first thing that we found was that the proteins that can generate this curvature can spatially localize in the cell membrane, and the way they do that is that they localize to the area of high curvature, which happens in these tubules. So this is a model generated plot of it, and this is experimental measurement, so you can actually see the similarity. But more importantly, the formation of these structures is actually dependent on the tension that you have on the cell membrane, which is connecting it back to the mechanics. So is this just a model prediction, or does this actually happen in real cells, right? So here I'm showing you comparison between experiments done using very high performance microscopy and our model, and you can see that we get all of the statistics of how many protrusions that we get and how many proteins are there. All of those comparisons actually come out, which essentially tell you that these types of mechanisms are actually what is driving the cell phenotype. So if this were really true, then the argument will be that the same proteins that are causing these curvature are the proteins that are actually controlling cell survival, and the difference between this 2D and 3D. And so if we then knock out these proteins, then we should be able to eliminate the difference between 2D and 3D. So that was the prediction, and then we actually go and do that experiment, and we find that exactly that's what happens, right? So so we can close the loop, right? You first do the prediction using these sophisticated mathematical models, and then you find a mechanism, and then you go back and validate that back in the cellular environment. So this was quite intriguing to us, and so what we then wanted to do is to ask, so can we now ask how do these exosomes get generated, and why are they mechanically coupled, right? So here I'm showing you some simulations that tell you under certain conditions, when you have the cytoskeleton, you can spontaneously generate these types of vesicular structures, and we can actually then tell, oh, what is it that you need to actually get these structures, right? So you need a certain cytoskeletal density and a certain what we call actin cortical tension, and if you program both of those in a unique space, then these types of vesicles get generated. And so you can produce them either using cytoskeletal mechanisms, or you can produce them using mechanisms that involve glycoproteins that accumulate on the cancer cell surface. Either way, you can get these types of structures, and this is what we manifest themselves as exosomes, kind of experimentally. And it turns out that there are underlying cellular processes that can accelerate this, and these usually involve these proteins called Rab GTPases. And I'm not going to get into the details, but this is what links the mechanics. So the extracellular matrix usually densifies when the tissue stiffness, and that will then cause a set of proteins on the cell membrane called the integrins to activate, and then the integrins will then activate these RABs, which will then produce more exosomes. And if you put that all in a mathematical model, you can show that this has like a switch-like response. It's almost as though, for small stiffness, nothing ever happens, and then you flip a switch, and then, once the stiffness increases in the tissue, these exosomes start generating, and that's essentially the prediction in the model, and that's exactly what you see in the experiment. But more importantly, if you now take these exosomes from patient sample cells or even from mice, then you can show that the exosomes that are grown in stiff tumors, in stiff conditions, can actually accelerate the production of larger tumors. So this tells you that these mechanically stimulated exosomes have this oncogenic potential and actually then accelerate the tumor growth. So that kind of brings me to the conclusion. This is one application of a fairly sophisticated set of models to kind of get at the mechanism underlying process that drives cancer that was otherwise not clear by just doing experiments, right? So this is an example of how mathematical models can actually help gain that insight. So the conclusions, what we learned from this is, of course, is that the cell survival is fundamentally governed by dimensionality, which means that it really depends on what the tissue architecture is. And the cell survival, as well as the extracellular vesicle production, are linked to the mechanics of the tissue and also drive mechanosensitive pathways. So this is a very unique finding because this nobody has thought about mechanosensitivity in this fashion before. And of course, the other obvious conclusion is that we can actually build now these biophysical models that can actually help explain these types of mechanisms. So more broadly, we've been using these types of models to understand other processes. Here I'm showing you an example of using this for nanomaterial applications. So these exosomes are nanoparticles that are naturally produced in the body. They're 100 nanometers in size. Other naturally occurring nanoparticles are viruses. But we can also engineer nanoparticles of polymeric particles or liposomes. But all of these can be studied using the same mechanism of how these particles interact with the cell membrane. And so we are working on several projects that involve the interaction of these nanoparticles, stiff nanoparticles, tethered nanoparticles, as well as flexible nanoparticles, and how they interact with the cells, but how they also are uptaken by the cells. So these are things that we can study using the same methodology. And this has a lot of relevance to the acute respiratory distress syndrome, which is a particular kind of infection that happens in severe SARS-CoV-2 patients. So we've been trying to understand why coronavirus, which is a soft virus, is much more capable of infecting a patient with ARDS compared to, say, a crystalline virus, like, say, an adenovirus. And so ultimately, it comes back to these types of mechanical pathways and this mechanotransduction, which we have highlighted here. But I would like to end, of course, on a broader kind of note. And on the computational side, I think it's important to emphasize that these models are, of course, multiscale in nature, but also both in terms of machine learning methods, but also into how powerful our computers get in the high-performance computing world. We are at a very exciting juncture because there is tremendous explosion of methodologies and technologies on both these aspects. And there is unbound opportunities for future scientists and students to actually combine them. And so one of the goals of my lab is to train my students in all three technologies, namely multiphysics modeling, machine learning, as well as high-performance computing. Of course, you can read more about this on our website. So with that, I'd like to kind of conclude by essentially, again, acknowledging that I have the privilege of working with very interdisciplinary and diverse teams in my lab. And a lot of the ideas that I spoke about today came from my students and my collaborators. And I thank them for being part of the lab. And I thank you for your attention.